Welcome to the deep dive. We're here to get into the crucial research, the stuff you really need to know, but make it quick and clear.
That's the plan.
So today, we're looking at a tool that gets talked about a lot in conservation circles, environmental DNA, eDNA. And the big question isn't really, does it work anymore? We know it does. The real question is, how well? And how do we make sure it works when it absolutely has to?
Exactly. eDNA often gets painted as this sort of magic wand. Wave it over the water and boom, you know what's there.
Right. Effortless detection.
Yeah, but for really high-stakes stuff like biosecurity, stopping a nasty invasive species, you can't rely on a tool if you don't know precisely how accurate it is under different conditions. Guessing isn't good enough. We need numbers.
Which brings us to our mission for this deep dive. We're getting into the paper, Quantifying the Sensitivity of Targeted eDNA Surveys to Improve Detection of Invasive Cane Toads, Rhinella marina. The lead author is Lawler.
And this study, it's really important because it gives us that quantitative manual. It helps turn eDNA from just a cool technology into something more like a reliable forensic tool.
We're going to show you that the effectiveness of eDNA, what scientists call its sensitivity, basically the chance of finding something if it's actually there. It's not just one fixed number.
Not at all. And the beauty of this research is that it lets scientists figure out exactly how much sampling effort they need, site by site, to be confident, say 95% confident that they'd find the target if it was present. It's like personalized sampling design.
Okay, let's unpack the why. Why is this so critical? The paper uses cane toads as the case study. Maybe give us the quick rundown on them. Introduced in 1935.
Right, 1935 in Australia. And they're big toxic amphibians. And they are marching across northern Australia, the Kimberley region specifically, at this alarming rate, about 50 kilometers a year.
That's incredibly fast. And the impact?
Oh, it's devastating. Native predators, snakes, lizards, quolls, they try to eat the toads and die because the toads are so poisonous. It leads to these massive population crashes and just throws entire ecosystems out of whack.
So if you're trying to manage this, trying to stop the spread,
yeah,
the absolute key is finding them early, right?
Spot on. You need to detect them right when they first arrive in a new place, a new water body, when there aren't many of them yet. That's when you need your detection method to be super sensitive, maximum sensitivity required.
And that's where eDNA comes in, isn't it?
Exactly. It's the obvious tool for the job. Targeted eDNA surveys, they can be incredibly sensitive. And importantly, much more cost-effective than sending teams out to visually search huge, often really difficult swampy areas.
But you mentioned a gap earlier. People were excited about eDNA, but what was missing before this study?
What was missing was the hard data, the actual number for absolute sensitivity in real-world conditions, out in the field where things are muddy, messy, variable.
Right. Not just in a clean lab setting.
Precisely. If we don't understand what environmental factors turn the sensitivity up or down like a dimmer switch, we could be spending a lot of money on surveys that don't actually give us much certainty.
So this paper set out to quantify those dimmers.
Yes. Things like, how many toads are there? How big is the water body? How much water could you actually filter? And what life stage are the toads in?
Okay, so how did they go about getting these numbers? This wasn't a lab simulation, you said.
No, definitely not. They went straight to the invasion front, West Kimberley, Western Australia. They sampled 35 water bodies, wetlands, farm dams, places toads really rely on during the dry season, which was when they sampled June, July 2023.
Because water is scarce then, so the toads congregate.
Exactly, concentrates them. And they also had 5 control sites confirmed toad-free, way ahead of the known invasion zone.
And they used 2 methods side by side, the old way and the new way.
Yep. For the old way, the conventional visual surveys, they did nighttime searches, walking around the edge of the water, spotlights, counting toads. They took that raw count and turned it into an encounter rate, basically, toads seen per minute of searching. That became their proxy for toad density at each site.
And the eDNA side. What was the sampling strategy there?
They took 5 water samples from around the edge of each water body, spaced out evenly, aiming for 500 milliliters each time.
Aiming being the key word.
Yes. Because here's the real world kicking in. The water was often really turbid, full of sediment, algae, stuff that clogs filters fast. So the actual amount of water they could get through the filter varied hugely. Sometimes they got the full 500 bills, sometimes barely 15.
Wow. But that difficulty, that variation, It actually turned into useful data, didn't it?
It absolutely did. That variable filtered volume became one of the key factors they tested in their model. It showed directly how water clarity, or a lack thereof, impacts your chances.
But hang on, if a sample comes back negative for toad DNA, how do you know why? Did you just fail to collect any DNA floating around, or did something go wrong back in the lab when you tried to test the sample?
That's a fantastic question. It's the crucial distinction. Is the failure in the field or in the lab? Like, did the mail carrier never pick up your letter, or did the post office lose it?
Good analogy. So how did they tease those two things apart?
They used a pretty sophisticated statistical method called a hierarchical occupancy model.
Okay, break that down a bit.
What it essentially lets you do is estimate two different probabilities separately. First, the field sensitivity. What's the chance you actually captured toad eDNA in your water sample, given it was there? And second, the laboratory sensitivity. Given the DNA was in your sample tube, what's the chance the lab process successfully detected and amplified it?
Amplification. That's the qPCR step, right?
Yeah.
Quantitative polymerase chain reaction, making lots of copies of the DNA target.
Exactly. qPCR is how you detect and quantify the DNA. So this model cleverly separates the capture step in the field from the detection step in the lab.
Which means they could isolate how those field conditions, pond size, toad numbers, water clarity, that sort of thing, specifically affected the capture part.
Precisely. Independent of how well the lab machines were working that day. And just on the lab side, they were rigorous. All 74 potential positive hits were double-checked using Sanger sequencing, and all their negative controls came back clean. So the lab side was solid.
Okay, super rigorous. So the big reveal. What did this model show? You said overall sensitivity was high, like 90%?
Right. Yeah, the average absolute sensitivity was around 90%, which is great. But the key finding was how much it varied. Site to site, it ranged from as low as 38% all the way up to 100%.
A massive range. What were the dimmer switches? What factors drove that variation?
The model pinpointed 4 key things that significantly influenced the probability of capturing eDNA in a sample.
Let's tick them off.
Okay, first one, no huge surprise. Toad density. The higher the encounter rate, the more toads seen per minute, the higher the eDNA sensitivity. More toads shedding DNA, easier to find.
Makes sense. Number 2.
Sample volume. That difference between struggling to filter 15 milliliters versus getting a full 500 milliliters, it mattered a lot. More water filtered equals a better chance of catching DNA. Simple as that, but now quantified.
Okay. Third factor was about the place itself.
Yep. Water body size, specifically the perimeter length. Sensitivity actually decreased as the perimeter got larger.
Ah, the dilution effect.
Exactly. Same amount of DNA potentially shed, but spread out over a much larger edge. The average perimeter was about 200 meters, but some were over 500 meters. Makes it harder to stumble across the DNA when sampling.
And the fourth factor, you said this one had the biggest effect, the real surprise.
Tadpoles. The presence or absence of cane toad tadpoles. This was the game changer.
Really? Tadpoles?
Yeah. If tadpoles were present at a site, the average probability of amplifying the DNA in the that lab sensitivity part was 0.80, so 80%. But if tadpoles were absent, that probability dropped way down to 0.34, only 34%.
Wow, that's a huge difference. Why would the little tadpoles matter more than the big adult toads for detection?
It likely comes down to their biology and behavior. Tadpoles are totally aquatic, right? They live in the water 247. They're constantly shedding skin cells, mucus, waste, all containing DNA directly into the water.
Okay.
And they often hang out in large groups, aggregated in the shallow edges, exactly where people tend to collect water samples. An adult toad might just visit the water's edge sometimes, maybe sit in it for a bit. But the tadpoles are like continuous, concentrated DNA dispensers right where you're sampling.
That's fascinating and incredibly useful for planning surveys. So looking at the overall picture, eDNA versus visual surveys, they both found toads at 23 sites, right? So equal footing.
Well, yes and no. They detected the same number of occupied sites, 23 each, but critically, they didn't detect the exact same 23 sites.
Oh, okay.
There were 4 sites where the visual surveys found toads, but eDNA missed them. And importantly, there were other 4 sites where eDNA found toads, but the visual surveys missed them.
So they complement each other. Each method has strengths and can pick up things the other misses.
Precisely. They're complementary tools, not necessarily replacements for each other in every situation.
So what does all this quantification, all these numbers actually mean for the conservation manager, the person on the ground trying to stop the toads? How does this change things?
It changes things significantly because it allows for strategic tailoring of the survey effort. No more one-size-fits-all sampling.
Right.
You can now look at a specific water body, assess its characteristics. How big is it? How murky is the water? Did we see tadpoles? And then use the relationships quantified in this study to calculate how many samples you actually need to take to reach your desired level of confidence. Say that 95% detection probability.
So if you roll up to a pond and it's full of tadpoles.
That's basically your easy mode for eDNA detection. The study showed that when tadpoles were present, you could achieve a 95% survey sensitivity with just 1 or maybe 2 water samples.
Wow. Regardless of other factors.
Pretty much, regardless of how big the pond was or even the adult toad density. That finding alone could save enormous amounts of time and resources. You sample, check for tadpoles, and if they're there, you might need far fewer samples than you initially planned.
Okay, but let's talk about the flip side. The tough scenario. The one where early detection matters most. A big, murky pond with only a few adult toads just arriving.
That's the critical challenge, isn't it? The early invasion, low-density situation. The model let them explore that worst-case scenario.
What did it predict?
Okay, picture this. A site with low toad density, say, equivalent to seeing 1 toad per minute of searching. A large perimeter 500 meters around, and really turbid water, so you can only filter maybe 10 milliliters per sample before the filter clogs solid.
The nightmare site.
The nightmare site. To achieve 95% confidence of detection in that scenario, the model estimated you would need to collect and process, wait for it, 69 water samples.
69. Okay, let's put that in context. Standard eDNA protocols often use, what, 5 samples? Maybe 10 per site.
Exactly. 5 to 10 is pretty typical.
So going from 5 samples to nearly 70? That's not just a small increase. If you're planning surveys across dozens of sites, that difference completely changes the budget, the logistics, the feasibility. It goes from a field trip to a massive operation.
That's the power of this quantification. Sensitivity isn't free. It has a quantifiable cost in terms of sampling effort. And now agencies can make informed choices. Is this high-value conservation area worth the cost of 69 samples? Maybe yes. Is this lower priority dam? Maybe the cost is too high and resources are better spent elsewhere.
So it sounds like the smartest approach is probably using both methods strategically.
Absolutely. A combined or phased approach makes a lot of sense. Start with the relatively cheap and quick visual surveys. They work well for big, obvious things like adult cane toads, especially if densities are reasonable.
Fly in the easy ones first.
Right. And then you deploy the more intensive, tailored eDNA sampling specifically at those sites where the visual surveys failed, or in really high-priority areas that absolutely must be kept toad-free, where you need that maximum confidence.
Now, before we wrap up, you mentioned the model wasn't perfect. The authors acknowledged some limitations, right? Some remaining mysteries.
They did, and it's important. There were 2 sites where adult toads were actually quite abundant, visually confirmed, and where the model predicted eDNA sensitivity should have been high. But the eDNA surveys failed to detect them.
So something else was going on.
It strongly suggests there are other environmental factors, things not included in this specific model, that can sometimes interfere with eDNA detection.
Like what sort of factors?
Things that might degrade DNA faster in the water, like high UV light exposure, extreme temperatures, or certain pH levels, or things that actively inhibit the qPCR process back in the lab chemicals, like humic acids from decaying plants, or maybe high concentrations of algae or sediment physically interfering.
So future research needs to try and incorporate those variables too.
Definitely. Building those environmental inhibitors and degradation factors into future models will likely explain that remaining variation and make the predictions even more accurate, even more robust.
Which brings us to the big picture, the final thought here. What Lawler and the team have really shown us, it seems, isn't just that eDNA can find species. It's that we finally have the quantitative tools, through this kind of careful analysis, to figure out how likely we are to find them in any given situation.
That's it, exactly. We can assess the feasibility, calculate the effort needed, and actually work towards guaranteeing detection with a specified level of confidence.
Whether you're tracking cane toads, or maybe a much rarer, more cryptic species you care about, platypus maybe, or endangered fish like the large-toothed sawfish.
Yes, the principle applies broadly. This research provides the framework. We can move beyond just hoping our standard sampling protocol is good enough. We can actually adapt our sampling intensity based on the unique conditions of density, the habitat size, the water quality, the life stage of every single place you might survey. That's how you ensure your conservation goals are met with real, quantifiable confidence.
A really fantastic deep dive. So the key takeaway for you listening, eDNA sensitivity. It's not fixed. Think of it like a dial, not an on-off switch. And the main things controlling that dial are tadpoles, how much water you filter, how many animals are around, and how big the place is.
Knowing that lets us be much smarter about how we use this powerful tool.
It means we can finally tune our efforts to make early detection truly reliable. Thanks for joining us on the Deep Dive.